Welcome to Generating AI Futures in Healthcare and BioPharma. Today we're doing a real deep dive into the world of cancer genomics. We're focusing specifically on a pretty ingenious AI tool from Google Research called DeepSomatic.
Yeah, this one's really making waves. It's potentially a massive step forward for precision medicine.
So our mission today is to really pull it apart, right? Look at the tech, the architecture.
Definitely. We'll get into the technical nuts and bolts, but also the groundbreaking data set they actually had to build just to train this thing. That's a huge story in itself.
And why it's setting a new benchmark for accuracy.
Exactly. Especially for finding those tiny, tiny genetic changes that actually drive cancer. Small variants, SNVs, indels, the tricky stuff.
Okay, let's set the stage, the core problem. It sounds simple, but it's not. Cancer is fundamentally a genomic disease.
Right, that's the bedrock. What it means is a typical tumor isn't just one thing gone wrong. It's riddled with thousands, sometimes tens of thousands of individual genetic changes, somatic variants.
And these variants aren't neatly organized. They're scattered across this. You called it a mosaic cell population.
Yeah, a mix of different cell clones within the tumor, each potentially carrying slightly different mutations. Plus, you got normal cells mixed in. It's messy.
So these aren't the variants you inherit from your parents.
No, that's the key distinction. Inherited variants, the germline ones, are in every cell. They're relatively easy to spot. These somatic variants are acquired after birth. Think environmental damage, UV light, smoking, or just random errors when cells copy their DNA.
And they're only in the tumor cells.
Or a subset of them, which makes finding them, especially the really small ones, single base changes, small insertions or deletions, indels, incredibly difficult. Like finding needles in a haystack made of slightly different needles.
So DeepSomatic is trying to pinpoint these specific culprits hiding in all that biological and technical noise.
Precisely. It builds on deep variant, which was already very successful for germline variants.
Right, we covered deep variant before.
But DeepSomatic is heavily modified. It had to be, to handle the specific complexities of cancer genetics, the heterogeneity, the low frequencies, the artifacts.
And the payoff if it works.
Huge. If we can reliably find the exact somatic variants driving a specific patient's cancer, well, that tells doctors which targeted therapy might work. Precision oncology.
Or it could point the way to completely new drugs.
Absolutely. It accelerates that whole pipeline from diagnosis to drug development. It's a really critical piece of the puzzle.
Okay, let's really hammer home that fundamental difference DeepSomatic has to deal with compared to its predecessor, deep variant.
Right. It boils down to timing and location. Germline variants, the ones deep variant handles, you inherit them. They're in basically every cell, and they're generally stable throughout your life. We kind of know what we're looking for.
That's somatic variants.
Acquired. Post-fertilization. Caused by external factors like carcinogens or internal ones like simple errors in DNA replication during cell division. And crucially, they are only in the tumor cells, or some fraction of them.
So the signal is much weaker, much more localized.
Exactly. And that immediately creates problems when you actually try to sequence a tumor sample in the lab.
Because you never get just pure tumor cells, do you?
Almost never. You get this mix. Tumor cells, yes, but those tumor cells themselves aren't uniform. That's the heterogeneity. They have different variants at different frequencies.
Okay.
And mixed in with those diverse tumor cells, you have normal, healthy cells. The amount varies sample to sample. That's the tumor purity issue.
So heterogeneity within the tumor and varying purity of the sample itself.
Yeah.
How does that actually mess up the detection process? Let's make it concrete.
Okay, imagine you're sequencing DNA from a specific spot in the genome. You get, say, 100 sequence reads covering that spot.
Right.
Now, if a crucial cancer-driving somatic variant is only present in maybe 10% of the tumor cells,
Okay.
and your tumor sample is only 50% pure, meaning half the DNA is from normal cells,
Right. Okay, so the math.
that variant might only show up in 5 out of those 100 reads. That's a variant allele frequency, or VAF, of just 0.05 or 5%. It's a tiny signal.
And the sequencing machines themselves, they're not perfect, are they? They make errors.
Exactly. Even the best machines have an inherent error rate. Maybe it's, say, 0.5% or 1%. So the machine might just randomly misread a base, creating what looks like a variant.
So how do you tell if that 5% signal you saw is a real biologically significant somatic mutation, or if it's just background noise from the sequencer?
That's the million-dollar question, and that's where traditional statistical methods really start to struggle. They look at the frequency, they look at base quality scores, and they try to calculate a probability.
But if the real signal is down near the noise floor,
It gets very fuzzy. They make mistakes. They call false positives flagging errors as real variants, or they miss real variants false negatives because the signal is too weak compared to their thresholds.
and you mentioned normal and tumor contamination, TIN.
Right, another wrinkle. Sometimes the normal tissue in the sample isn't perfectly normal and might carry some low-level artifacts or even very low-frequency germline variants that can confuse the picture further, looking like somatic changes. DeepSomatic needs to filter all that out.
So it's not just about finding a difference, it's about figuring out if that difference is a real somatic event or just junk. Technical artifacts. Are these artifacts a big deal?
Huge. And critically, they're often not random. The sequencing process itself, the chemistry, the optics, the alignment algorithms, they introduce systematic errors, predictable patterns of mistakes.
Predictable. That sounds like something AI could learn.
Exactly. Traditional methods often see these patterns like a specific type of error always happening near the end of a read and flag it as a potential low-frequency variant because, statistically, it looks like one. DeepSomatic aims to learn the signature of these artifacts versus the signature of a true variant.
This gets even worse with certain common sample types, right? Like FFPE, formalin-fixed paraffin-embedded tissue.
Oh, absolutely. FFPE is vital for storing patient samples long-term in hospitals. It's how pathology works. But the formalin fixation process, it chemically damages the DNA.
In what way?
One of the most common types of damage is deamination of cytosine bases, which makes them look like thymine bases during sequencing, so you get these artificial C to T changes. And often this damage is worse at the ends of the DNA fragments generated during sample prep.
So a traditional caller sees a C to T change. Maybe it's low frequency, maybe it's near a read end.
And it might just say potential somatic variant, because statistically that's what the simple model sees.
But DeepSomatic.
Because it's looking at the whole picture, the image, it can see the C to T change and its low frequency and its position at the read end, and maybe correlated lower base quality scores typical of FFPE damage. It learns that whole constellation of features often means artifact, not mutation. It's pattern recognition on a much deeper level.
That makes sense. What about the difference between whole exome sequencing, WES, and whole genome sequencing, WGS?
Another key challenge. WGS sequences, well, pretty much the whole genome. WES is a shortcut. It targets just the protein coding regions, the exome, which is only about 1 to 2% of the genome.
Cheaper, faster.
Exactly. Very common clinically. But the process used to capture just those exome regions introduces its own unique biases and noise patterns into the sequencing data. It looks different from WGS data.
So a tool trained only on WGS might not work well on WES and vice versa.
Correct. They have different error profiles. A robust tool like DeepSomatic needs to be able to handle both, ideally with models specifically tuned for each.
Which brings us, inevitably, to the biggest bottleneck they faced before they could even start building these sophisticated models: the data, or lack thereof.
The data drought, absolutely. You can have the cleverest algorithm in the world, but supervised machine learning models are fundamentally data hungry. They need large, accurate, representative training data sets to learn from.
And for germline variants, Deep Variant had resources like GIE.
Right. Genome in a bottle. Millions of high-confidence, well-validated, inherited variants. A fantastic resource.
But for somatic variants, the cancer-specific ones.
It was sparse. Really sparse. Before DeepSomatic's efforts, the main public, high-quality, comprehensively validated benchmark dataset was the SEQC2 dataset, based on a single cell line pair, HCC 1395.
How many variants did that have?
Tens of thousands, which sounds like a lot, but compared to the millions in GIAB and considering the sheer diversity of mutations across different cancer types, it just wasn't enough.
Orders of magnitude less data.
Exactly. And that scarcity severely limited how accurate and, crucially, how generalizable previous somatic calling models could be. They might work okay on HCC 1395, but perform poorly on a different cancer type or data from a different sequencing machine.
So the Google research team knew.
They knew they couldn't just build a better algorithm. They had to build a better data set first. They had to solve the data problem before the modeling problem could even be properly tackled.
Okay, so they do the biological challenge, they do the data challenge. Now let's get into the machine itself. How does DeepSomatic actually work? You said it turns this into an image recognition problem.
That's the core idea, inherited and adapted from Deep Variant. Instead of relying on complex statistical formulas and arbitrary thresholds, which, as we said, often break down with low-frequency variance or pattern noise, DeepSomatic uses a convolutional neural network, a CNN.
The same kind of AI used for recognizing faces or objects in pictures.
Pretty much, yeah. It trains the CNN to visually identify the patterns in sequencing data that correspond to true somatic mutations versus patterns that indicate germline variants or sequencing errors.
Fascinating.
Yeah.
So how do you get from raw DNA reads, those BAM or CRAM files, to an image the CNN can understand? Let's walk through the pipeline, starting with that first step, make example somatic.
Right. This is the crucial data preparation and transformation stage. The script make example somatic takes three main inputs. You need the aligned reads from the tumor sample.
The BAM or CRAM file.
Yep. And the aligned reads from the matched normal sample, assuming you have one. We'll get to tumor-only later.
Okay, normal BAM CRAM.
And third, you need the reference genome sequence, usually a FASTA file, so it knows what the normal sequence is supposed to look like at any given position.
Got it. So what does make example somatic do with these?
It does two main things. First, it scans through the genome, looking for candidate variant sites. The basic logic is find positions where there's some evidence of a variant in the tumor reads, but not significant evidence of that same variant in the normal reads. It uses some initial thresholds just to screen possibilities.
So it's filtering down the possibilities first.
Exactly. You don't want to generate an image for every single base in the genome. Once it finds a candidate site, it generates this special data structure, technically a tensor, but we often call it a pileup image.
The pileup image. Tell me more about that. What does it actually look like? This sounds key to how DeepSomatic leverages both samples.
It really is. Imagine you take all the individual sequence reads that cover that candidate variant position in the genome. The script stacks them up visually. Crucially, reads from the normal sample are put in the top half of this image or tensor.
Okay, normal on top.
And reads from the tumor sample are put in the bottom half.
Tumor on the bottom. So the CNN literally sees them side by side.
Precisely. It provides immediate visual context. The CNN can directly compare what does the inherited DNA look like here versus what does the potentially mutated tumor DNA look like here. It's not just subtracting one from the other statistically. It's a direct visual comparison.
That's clever. But it's not just a simple black and white image of A's, T's, C's, and G's, is it? You mentioned multiple channels, like a detailed map.
Right. It's much richer than that. This is where the deep learning power comes in. The tensor has multiple channels, each encoding different kinds of information about the reads at that position. The source paper mentions 5 main categories feeding into these channels.
What kind of information are we talking about?
Okay, so channel 1 might represent the read base itself, A, T, C, G, or maybe in insertion deletion. That's kind of the basic color map.
Makes sense.
Channel 2 could be the read base quality score for each base. This tells the CNN how confident the sequencing machine was about that specific letter. If a potential variant only appears on bases with terrible quality schools, the CNN learns that's likely noise.
Like a confidence layer.
Okay. Then you have channels related to alignment. Channel 3 might encode mapping quality. How confidently was the entire read placed at this location in the genome? Reads mapping poorly, maybe in repetitive regions, often cause false variant calls.
So the CNN sees the variant and knows the reads supporting it are poorly mapped.
Exactly. It learns that correlation. Other channels capture things like whether the read mapped to the forward or reverse strand, details about the alignment itself, like clipping, maybe information about insertions or deletions nearby. By layering all this data, the complex molecular information is translated into a rich visual pattern that the CNN is trained to interpret.
Okay, so make example somatic creates these rich multi-channel images comparing normal and tumor reads at candidate sites. That sets up step 2.
Yeah.
Call variants. This is where the CNN does its work.
This is the classification engine. The call variant script feeds these image tensors into the pre-trained convolutional neural network.
And this is where DeepSomatic differs significantly from standard deep variance goal.
Yes, remember, deep variant is primarily doing genotyping, figuring out if a site is homozygous reference 00, heterozygous error 1, or homozygous alternate 11 based on inherited alleles. DeepSomatic's CNN is trained for a different task, cancer-specific classification.
It's deciding the origin story of the variant.
That's a great way to put it. Based on the patterns in the pileup image tensor, the CNN outputs a classification for that candidate site. It decides, Is this likely just the reference sequence, maybe with some sequencing noise? Is it a germline variant inherited present in the normal sample too? Or the crucial one, is it a true acquired cancer-associated somatic variant?
And because it's learned from those complex images, it can make that call, even with low VAFs or tricky artifact patterns.
That's the whole point. It learns the subtle visual signatures that distinguish real somatic events from germline variants and from those structured sequencing errors like the FFPE CT artifacts we talked about.
Okay, the CNN makes its call. Then step 3, post-process variants, getting the results out in a usable format.
Right. This final script takes the raw predictions and schools from the CNN and formats them into the standard variant call format, the VCF file, or its genomic counterpart, GVCF. This is the file researchers and clinicians actually work with.
And critically, how does DeepSomatic communicate the CNN's classification in that VCF file? It uses the filter field, right?
Yeah. Yes, the filter column in the VCF is key. Instead of just giving a variant and a quality score, DeepSomatic uses specific filter tags defined in the VCF header to tell you what the model decided.
Let's look at the example from the documentation. If I see a line of the VCF and the filter column says pass, what does that signify?
Pass means the model is confident that this is a true somatic variant. All internal quality checks and classification criteria were met. This is the signal you're usually most interested in for cancer research or treatment decisions.
Okay. And what if it says germline?
Germline means the model classified it as a non-somatic variant, likely inherited, present significantly in the normal sample as well. Important to know, but probably not a direct cancer driver you'd target.
And refcall?
Refcall means the model decided the site is actually a homozygous reference. No variant here, just noise or maybe extremely low-level stuff below the calling threshold. And low qual means a potential variant was seen, but the model's confidence score was below the threshold needed to call it pass or germline.
So looking at their example output, tree R1, 14,003, CG, 43.8, pass S, VAF.1.
Yep, that tells you. Chromosome 1, position 14,003, reference is C, variant is G, quality score 43.8. Critically, filter is pass, meaning DeepSomatic calls this a confident somatic variant. The additional info shows the VAF is 1 or 100% in this case, meaning all tumor reads showed the G.
But you could also have a pass variant with a much lower VAF, say 0.05.
Absolutely. And that's the power. The CNN, having seen the full context in the image tensor, might still confidently call it pass S, even at 5% VAF, because the pattern looked right. Maybe clean reads, good mapping quality, clear difference from the normal sample image, whereas a simple statistical caller might have dismissed it as noise or tagged it low call.
So the architecture translates this incredibly complex biological and sequencing problem into a format where deep learning can effectively separate signal from noise.
That's the essence of it. Reads to images, images to classification, classification to clear VCF output.
That architecture sounds incredibly powerful. But we keep coming back to this point. It's only as good as the data it learns from. And the existing somatic data was lacking.
Severely lacking. They knew this. Relying only on that single SC2C2 HCC1395 dataset just wouldn't cut it. A model trained on only one breast cancer cell line wouldn't generalize well. It wouldn't understand the quirks of lung cancer mutations or different sequencing platforms or different sample preps like FFPE.
So they had to build their own training resource. This is the Castle dataset you mentioned, Cancer Standards Long-Read Evaluation.
Exactly. And this wasn't just about getting more data. It was about getting better, more diverse, and more reliably validated data. They needed a true gold standard.
How did they approach building it?
Very methodically, they selected 5 matched tumor-normal pairs of well-characterized cancer cell lines.
Cell lines, not patient samples. Why?
Cell lines provide a consistent, renewable source of DNA. Patient samples are variable and finite. For building a benchmark and training models, cell lines give you stability. They used the existing HCC1395 breast cancer line and added 3 more breast cancer lines, HCC937, HCC1954 plus 2 lung cancer lines, H1437 and H2009. This immediately introduced more cancer type diversity.
And they were careful about the source material.
Extremely. For 4 of the 5 pairs, the tumor and normal DNA came from the exact same cell culture extract. This is subtle but important. It minimizes the chance that tiny differences in cell culture conditions could create artificial variants, ensuring the somatic variants they found were more likely to be real.
Very rigorous. Now, the really novel part seems to be the multi-platform sequencing approach. They didn't just use Illumina.
No, and this was key to creating the high-confidence truth set. They performed whole genome sequencing, WGS, on all 5 cell line pairs using 3 major sequencing technologies simultaneously.
3. Which ones?
Illumina, the standard for short-read sequencing. PacBio Hi-Fi, known for its highly accurate long reads. And Oxford Nanopore Technologies, or ONT, famous for very long reads, even if the per-base accuracy is traditionally a bit lower than Hi-Fi.
Why use all 3? Isn't that expensive and complex?
It is, but it was crucial for validation. Every sequencing technology has its own biases, its own characteristic error patterns. A variant called only by Illumina might be an Illumina-specific artifact. A variant only seen by ONT might be an ONT error.
So if a potential variant shows up independently on 2 or 3 different technologies...
And you can be much, much more confident that it's a real biological variant, not just a technological glitch. That was the core principle for building the Castle truth set. Only variants confirmed by at least 2 independent sequencing technologies or calling methods were included.
A cross-validation using the technologies themselves.
Exactly. It filters out the platform-specific noise automatically, leaving you with a much cleaner, more reliable set of true somatic variants to train and benchmark against.
And they didn't skimp on the sequencing depth either, right?
No, they went deep. For PacBio Hi-Fi, around 60x coverage, which is great for accuracy. For ONT, they went even deeper, about 90x coverage for the tumor, and 30x for the normal, leveraging the power of very long reads to resolve complex regions, even with slightly lower per-base accuracy. And standard high-coverage Illumina, too. Training models on data from all 3 forces the AI to become robust to different read lengths, error profiles, and alignment challenges.
Did this diverse dataset actually capture different biological signatures of cancer?
Yes, absolutely. When they analyzed the mutational patterns, they found clear differences. For example, those 2 lung cancer cell lines, H1437 and H2009, showed a strong enrichment for mutations associated with the SBS4 signature.
SBS4, isn't that the one linked to tobacco smoke?
Precisely. So the model wasn't just learning generic mutations, it was learning the specific kinds of damage associated with known cancer causes reflected in these different cell lines. That's vital for building a model that works in the real world.
Okay, the proof is in the pudding. What was the actual measurable impact of training on this richer multi-platform Castle dataset compared to just the old SEQC2 data?
The impact was massive. First, the sheer number of high-confidence benchmark variants available for training and testing increased by 6.5 times. They added over 263,000 new validated variants to the pool.
6.5 times more data.
And the performance boost was immediate. Across all 3 technologies, the overall error rate, combining false positives and false negatives for calling SNVs, single nucleotide variants, dropped by nearly 39%.
Almost 40% fewer errors for SNVs. That's substantial. But you hinted the indel improvement was even more dramatic.
Off the charts. For insertions and deletions, indels, the total error rate plummeted by 95.2%.
Wow, 95% reduction in indel errors. Why are indels so much harder to get right usually?
Indels are tough because they change the length of the sequence. When you're trying to align short sequencing reads back to the reference genome, an insertion or deletion creates a gap or an overlap that confuses the alignment algorithms, especially in repetitive DNA regions where indels often occur.
So statistical callers just get lost.
They often do. They struggle to distinguish a real small indel from alignment artifacts or polymerase slippage during sequencing. The fact that DeepSomatic CNN, trained on the high-quality, multi-platform Castle data, could slash indel errors by 95%. It shows that the image-based approach is capturing the complex visual patterns around indels, the alignment signals, the read support, the quality scores, in a way that statistical models just fundamentally can't.
So the data revolution enabled the algorithmic potential.
Absolutely. Castle provided the ground truth needed for the CNN to learn robustly, especially for those previously intractable indel calls. It proved that diverse, high-quality, multi-technology data was the key to unlocking the next level of accuracy. So, armed with the Castle dataset, DeepSomatic didn't just improve on previous benchmarks, it significantly outperformed the existing state-of-the-art tools. We're talking about established callers like Strelka2, Mutek2, Somatic Sniper, and the more recent deep learning contender, Claire's.
And the standout was really that indel performance. Let's put some numbers on that comparison again.
Yeah, the indel leap was remarkable. For standard Illumina WGS data, DeepSomatic hit F1 scores around 90% for indels. The next best tool was hovering around 80%. A solid jump.
Okay, 80 to 90%. Good.
But look at the long-read data, like PacBio Hi-Fi. Before DeepSomatic, the best F1 scores for PacBio indel calling were often below 50%.
Less than a crane flip chance of being right.
Pretty much. It was notoriously unreliable. DeepSomatic, trained on the Castle-Tac bio data, pushed that F1 score to over 80%.
Okay, wait, from under 50% to over 80%. That's not just an improvement. That fundamentally changes the utility of PacBio for indel detection in cancer, doesn't it?
Completely. It basically made reliable somatic indel discovery using PacBio data a reality for the first time. And since many important cancer genes can be disrupted by small indels, that's a huge clinical and research advancement.
Beyond just raw accuracy and benchmark data, though, they engineered DeepSomatic to be flexible for real-world clinical situations, right, where things aren't always perfect.
Exactly. A major hurdle in some clinical settings is the lack of a matched normal sample from the patient.
You mentioned pediatric leukemia as an example.
Right. For some cancers, especially certain blood cancers or when dealing with archival samples, getting that clean pre-cancer normal tissue sample just isn't feasible. So DeepSomatic needed a robust tumor-only calling mode.
How does that even work? If you don't have the normal baseline to compare against, how do you know if a variant is somatic or just an inherited germline variant?
It's definitely trickier. They developed specific tumor-only models for Illumina, PacBio, and ONT. The key is substituting the missing patient-specific normal information with population-level information.
How? What does it use instead?
In the make example step for tumor-only, it incorporates data from two main sources. First, a panel of normals, or PON. This is essentially a database built from sequencing many healthy individuals, cataloging common germline variants, and also common sequencing artifacts seen in normal samples.
Okay, so it knows what's common in the general population.
Right. And second, it uses population allele frequency databases like NOMAD or 1,000 genomes, which provide frequencies for known germline variants. This information gets encoded as additional channels in that pileup image tensor.
So the CNN learns to look at a variant in the tumor reads and ask, does this pattern look like something commonly seen in the panel of normals or population databases?
Exactly. If it matches a common germline variant signature from the PO or databases, the model learns to filter it out, assuming it's likely inherited, not somatic. It's not as precise as having the patient's own matched normal, but it's a very clever workaround.
Does tumor-only mode sacrifice much accuracy?
Generally, the precision might be slightly lower, meaning it might make a few more false positive somatic calls because it lacks that perfect individual baseline. But the recall, its ability to find the true somatic variants, remains quite high. It's excellent at discovery.
And this tumor-only capability had a direct, tangible impact in that leukemia study.
It really did. That collaboration with Children's Mercy Hospital was powerful. They took 8 pediatric leukemia samples that had already been analyzed using standard methods and ran them through DeepSomatic in tumor-only mode.
And the result?
DeepSomatic found all the known, previously identified cancer variants, validating its sensitivity. But crucially, it also uncovered 10 additional small variants, real somatic variants that the standard pipelines had completely missed.
10 new potential clues in kids' cancer samples. What does that mean practically?
It's potentially huge. Each of those missed variants could be a driver of the leukemia, maybe even an actionable target for a specific therapy. Standard methods miss them, perhaps due to low VAF, or being tricky indels, or noise from the tumor-only setting. DeepSomatic's ability to dig these out could lead to reevaluating treatment for those specific patients.
And on a larger scale.
It suggests there might be many more undiscovered low-frequency driver mutations hiding in existing cancer datasets. Finding them fuels the BioPharma pipeline, identifying new targets for drug development that were literally invisible before.
And DeepSomatic didn't just stop at standard WGS. It tackled those difficult sample types too. FFPE and WES.
Yes, they specifically developed and validated models for FFPE, WGS, FFPE, WESS, and standard WES. These are bread and butter sample types in clinical oncology and research archives. And again, the DeepSomatic models significantly outperform tools like Strata2 and Claris on these noisy, often lower quality samples.
Got an example for FFPE?
Sure. On FFPE whole genome data, DeepSomatic achieved an F1 score for SNVs around 0.88. The next best competitor was below 0.81. That difference matters when you're trying to reliably call variants from precious, chemically damaged archival tissues. It means potentially unlocking vast amounts of data from stored samples that were previously too challenging to analyze accurately.
Finally, the big test of any AI model, generalization. Did it work on cancers it wasn't explicitly trained on?
That's always a critical check. The main Castle training set used breast and lung cancer lines. So they tested the trained models on a completely different cancer type, a glioblastoma sample, an aggressive brain tumor. It worked. DeepSomatic successfully identified the known somatic variants present in that glioblastoma sample, even though it hadn't seen glioblastoma mutations during training.
So it learned general principles.
It suggests the model learned fundamental patterns of what somatic mutations look like versus germline variants versus sequencing artifacts, rather than just memorizing the specific quirks of the training cell lines. That ability to generalize is absolutely essential for a tool to be broadly useful.
The overall impact seems clear. For biopharma, for precision medicine.
It touches every stage, helping clinicians choose existing therapies by accurately identifying known driver mutations, highlighting new, previously undetectable variants that could become targets for entirely new drug classes. It just accelerates the whole cycle of discovery and application, pushing us closer to genuinely personalized cancer treatment based on the specific mutations in that patient's tumor. Hashtag tag outro.
Okay, let's wrap up this deep dive. Looking back, the success of DeepSomatic really feels like it hinges on two core pillars, doesn't it?
I think so. First, there's the clever deep learning architecture itself, that idea of transforming the aligned reads, both tumor and normal, into those information-rich pileup images.
Letting the CNN visually classify somatic versus germline versus noise.
Right. But that architecture, powerful as it is, could only reach its potential because of the second pillar, the data. The massive effort to build the Castle dataset.
That high-quality multi-cancer, multi-technology resource, using Illumina, PacBio, and ONT to get that cross-validated ground truth.
Exactly. That dataset allowed them to train models robust enough to handle real-world cancer complexity and achieve that step change in accuracy, especially for those notoriously difficult indels. It was a synergy of algorithm and data.
And importantly for the research community, they didn't keep this locked away. It's open source.
Fully open source. The tool itself, integrated within the well-maintained deep variant framework. All the key scripts we mentioned, run DeepSomatic, make example somatic, call variants, post-process variants are available.
And the Castle dataset too?
Yes, the dataset itself is available. They provide Docker containers for easy deployment. It's designed for the community to pick up and use, to build upon. It's a huge contribution to accelerate cancer research globally.
A fantastic example of open science in action.
Definitely. Which brings us to maybe a final thought to leave you with. DeepSomatic showed that by increasing the training data size and diversity, in this case by 6.5 times with Castle, they got these incredible performance gains, like that 95% reduction in indel errors.
Yeah, that number is sticking with me.
What it strongly suggests, I think, is that we are likely nowhere near the point of diminishing returns when it comes to training data for cancer genomics.
Meaning more data would likely lead to even better models.
Quite possibly, yes. Cancer is incredibly diverse. There are so many different mutational processes, different combinations of drivers, different tumor microenvironments. The known heterogeneity is vast. So while DeepSomatic is a huge leap forward, the next big frontier might not just be tweaking algorithms, but continuing the hard work of generating more high-quality, diverse, openly accessible sequencing data from different cancers, different patients, different treatments. Feeding these powerful AI models with even richer data seems essential to unlock the next level of truly personalized cancer insights.
The data challenge remains central. DeepSomatic has raised the bar significantly, showing what's possible when you combine cutting-edge AI with meticulously curated data. Thank you for joining us for this deep dive into generative AI futures in healthcare and biopharma. We'll see you next time.